my name is Tiara Padayachee, and today I will be presenting recently published results from my PhD studies with the following title. So globally in 2022, tuberculosis caused an estimated 1.3 million deaths, with approximately 7.5 million new cases being reported according to the World Health Organization. Approximately 410,000 people developed rifampicin-resistant or multi-drug resistant TB, highlighting the importance to develop new novel anti-TB drugs with a novel mode of action to combat this pathogen. The genome sequence of Mycobacterium tuberculosis revealed 20 cytochrome P50 monooxygenases, of which 3 could be anti-TB drugs, namely CYP121, 125, and 142. Gene knockout studies reveal that CYP121 is responsible or is essential for the viability of Mycobacterium tuberculosis. Cytochrome P50 monooxygenases are heme thiolate proteins that are found in all kingdoms of life. They are known to perform key enzymatic reactions in a regio and stereospecific manner, and thus become drug targets of interest when they are encoded into pathogens. The biological function of CYP121 was identified based on its downstream association with its RB2275 in an operon structure, in which the gene product is known to catalyze or produce two tyrosine residues in CYY, and CYP121 is known to perform the catalytic oxidative coupling of these residues, resulting in mycocyclosin. It is also able to hydroxylate a phenylaniline analog, which I'll be speaking about later on. So analysis of 53 crystal structures revealed that majority of them were in a closed conformation, which is expected as it is a drug target and therefore will be bound to numerous ligands. The average area and volume did decrease from open to closed conformation. However, the change is very minimal when compared to other P450s such as CYP107. RMSD of 0.225 was observed for 53 crystal structures, highlighting the rigidity of the structure. Active site analysis revealed various amino acids that are responsible for various processes, and mutations or manipulations of these amino acids further highlighted the roles that they played. For example, when serine 279 was mutated to alanine, the hydrogen bonding network associated with this protein was completely disrupted. The native substrate within CYP121 was analyzed, and there were various hydrophobic and hydrogen bonding interactions noticed. It's interesting to note that the 6-water ligand was not displaced upon binding. However, it did participate in the hydrogen bonding network that I mentioned previously. So this P450 does not follow the typical catalytic mechanism, as it does not generate an ion-bound oxidant. Instead, it performs a peroxidase type-like reaction. And so this study focused on the catalytic mechanism using percetic acid as an oxidant, and there were 4 different catalytic pathways. One of which you could have compound 1, as shown here, and there will be a hydrogen obstruction that will occur at the proximal tyrosine residue, resulting into a substrate radical in compound 2, and thereafter it could go through an electron tunneling step where there will be a second hydrogen obstruction that will occur at the distal tyrosine residue, and due to the opposite charges, they will then form the carbon-carbon bond. Or it could be a proton-coupled electron transfer step where it will migrate to the proximal tyrosine residue, and a second hydrogen obstruction will then occur, and tautomization will result in mycocyclosin. It could also form an oxyferryl species and a substrate radical, and thereafter follow the same steps, which is the electron tunneling and the proton-coupled electron transfer step. So the hydroxyl group of the native substrate was removed, and a phenylaniline analog was developed, as I mentioned before, and the catalytic mechanism was then reanalyzed. And so this study revealed that there was a loss of a methyl group and the production of the native substrate, as well as formaldehyde. So it resulted in clearly there was a carbon-oxygen bond cleavage via deformylation, as well as there was your oxygen demethylation reaction. So it's interesting to note that the hydroxyl group is required on the substrate for the carbon-carbon coupling to occur. So acyl drugs are known to interact with various P450s in which they are perpendicular to the heme and they directly coordinate to the heme ion directly. However, with fluconazole within this CYP121 revealed a novel mode of action in which it is able to incorporate the heme ion via a bridging water molecule. And this shows a novel binding mode. However, it's interesting to note that, remember, I highlighted the rigidity of the structure, and so structures in I helix like alanine and serine prevented the fluconazole from adopting the direct ligation mode, and so manipulation of these structures could possibly allow for fluconazole to have a direct coordination. So to understand the substrate specificities, this study focused on generating analogs of the native substrate, and here your diketopiperazine ring as well as your tyrosine side chains were modes of modification. And so they realized various amino acid interactions that are required for efficient binding, such as the hydrogen bond of the carbonyl group of the DKP ring and asparagine 85, van der Waals interaction with phenylalanine and tryptophan residues, the structure of the DKP ring, as well as the absence or incorrect position of the tyrosyl group. Other analogs included halogen and methyl analogs. It's interesting to note that in the halogen analogs, the iodine analog had a high binding affinity to CYP121, which could be due to its size and polarizability and having favorable interactions with the heme. The phenolic group was not essential for binding because no matter what you did with the phenolic group, whether it was blocked or whether it was replaced, it still had a binding affinity to its CYP121. So the next step that researchers then did was to develop potent inhibitors. So many studies focused on fragment-based approaches, which is a new technique that uses structural guidance in order to develop potent ligands from small molecular weight molecules, and they could be great chemical tools towards drug development. So in the first two figures, you can see fragments that were from a chemical library bound within CYP121. Fragment 14 had a high ligand binding efficiency and it directly coordinated to the heme ion, disrupting the hydrogen bonding network. The bottom two is retrofragmentation of a triphenylpyrazole amine compound, and this identified possible binding hotspots as well as critical interactions with active site residues. So the native substrate of this enzyme is Y-shaped, and therefore designing inhibitors of the similar shape could be a strategy to inhibit this enzyme. And so this study focused on developing that. And in the first one, we have your allyl derivatives of imidazole compounds. They did not interact with any active site residues, but they could possibly block the substrate access channel. And in the second one, we have an indole derivative as well as a pyridyl derivative, and it's a comparative analysis of the native substrate, fluconazole, and the 10J compound, which is the indole derivative, showed that they had either directly or indirectly interacted with 3 amino acid residues shown here. But hydrogen bonding with the glutamine and arginine 385 and 386 respectively could possibly successfully block the substrate access channel. So another strategy that researchers adopted was direct coordination with the 6-water ligand to disrupt the hydrogen bonding network. Here, I-47 was known to be a potent inhibitor. It consists of an imidazole moiety, a propylene linker, and a methylene bridge to join the aromatic residues. And here, L21 was of high importance as it directly coordinated to the heme ion, and it did show that it was able to inhibit CYP121 substrate conversion at certain levels. When you're looking at compound C or 6TE7, so in this compound, unfortunately, it was trapped in the distal active site due to a hydrogen bond network with a aspartic acid, and this was because a shorter linker bridge was used. So lastly, there were also tryptophan derivatives that were also tried to bound within CYP121. 10 and 21 were not used for further studies because they had a low ligand electron density. However, compound 14 which has an indole moiety, was able to result in the expulsion of many water molecules from the active site and therefore disrupted the hydrogen bonding network, which could be a novel binding mode distal to the heme. So what can we take away from this study? So firstly, CYP121 is highly substrate specific, it's highly rigid, and in terms of designing of inhibitors, you could use Y-shaped inhibitors, you could use inhibitors that will disrupt the hydrogen bonding network, either directly coordinating the heme. You could incorporate an indole moiety to result in the expulsion of water molecules, as well as incorporating a halogen molecule to have a higher binding affinity than the native substrate. So these results have been published in the International Journal of Molecular Sciences. I would like to thank my supervisor, Professor K. Saeed, the University of Zululand, and NRF for funding.